Hello and welcome to the Carolam Cymru revision sessions. Today is session 4 of this stint of revision sessions, and you can see what's been before. I covered plant and animal cells in session 1, and then you've had 2 sessions from Miss Abigail Kavanagh on heart and circulation and plants and photosynthesis, and it's for me to round these sessions off with cell transport, which I'm going to cover today. Worth noting, though, that we do have a live event on the 25th of October at 5:30, where you can ask any questions, and we'll also be there to sort of give an overview of what we have covered during these sessions as well. And if there's anything you want to ask, that'll be the time to do it. But let's get on with today's session. So today, like I say, we're looking at cell transport. And specifically, we're looking at these 3 types of transport or movement that occurs across membranes: diffusion, active transport, and osmosis. So these are the things we're going to try and cover in this session. So we'll start with the basics of diffusion, how it works, the significance of the cell membrane when it comes to diffusion. That is, whilst we can talk about diffusion happening sort of in a room, for example, really what we're really interested in biology is the movement of molecules across a membrane, and that's really what we're looking at with all 3 of these modes of transport. Then we're going to do the same for active transport, and we're going to then do the same for osmosis. So it's the diffusion of water, so it's an extension of that diffusion that we've covered already. And we will need to be referring to this idea of a selectively permeable or partially permeable membrane. Okay, so let's make a start with diffusion. Diffusion is something that you'll be familiar with, no doubt, either because you've studied it, but you're also familiar with the phenomenon. Because it's quite simple. Diffusion is just this spreading of particles from area of high concentration to an area of lower concentration. And you can see this image there sort of shows that in that top left glass, you can see some food dye has been squirted into this glass, and slowly those dye molecules distribute themselves around that glass. So in the bottom right, you've got those dye molecules more evenly distributed, and they started off at a high concentration when they were first put in, and slowly they moved or spread out through that glass to now be more evenly distributed. Same thing applies. I often use the example of, someone brings a bag of chips into a room, and that smell of chips spreads across the room. Now, whilst that's entirely correct for diffusion, we're really more interested in diffusion across membranes, diffusion that happens into and out of cells. But worth noting and worth getting to grips with the more basic idea of just diffusion within a solution, within water, but it can be within air. You start with a high concentration of molecules when that dye is put in, and those molecules spread out and they're more evenly distributed through that glass, or through that beaker in this case. When we have molecules moving from a high concentration to a low concentration, we have a difference in concentration between 2 areas. We have a high concentration area and a low concentration area. And when we've got that difference between 2 areas, we call it a concentration gradient, okay, like a slope going from the higher down to the lower. And we say then, when particles move from a high concentration to a lower concentration, we say they move down the concentration gradient from the high to the low. You can imagine that gradient or that slope moving from high to low, and they move down it, that concentration gradient. And that's very easy. It's very passive, meaning it doesn't require any energy. So we say it's a passive process because there is no input of energy. And it's worth noting that, because when we talk about active transport, we are going to see there is an input of energy, and there will be a reason for that. But diffusion is very straightforward. It's our starting point, really. It's a passive process. Passive is the opposite of the term active. Active means it needs energy. Passive means it doesn't. Right, 2 things that are key to us when I talk about diffusion are oxygen and carbon dioxide. So we think of oxygen, oxygen needed for respiration. Carbon dioxide is a waste product of respiration, but we could also say that carbon dioxide is needed by plant cells for photosynthesis. And it means there needs to be a movement of oxygen and carbon dioxide across a cell membrane, be it an animal cell membrane or a plant cell membrane. And there's a free passage of oxygen and carbon dioxide. So when we referred to the cell membrane before, we said that the cell membrane controls what comes in and out of the cell. But actually, it allows oxygen to move quite freely. There is no control there, and it allows carbon dioxide to move quite freely. And they will always move them from where they are at a high concentration to where they are at a low concentration. And it's showing it there. We have oxygen in the alveoli, so oxygen is in red, and there's a higher concentration of oxygen in the alveoli of the lungs and a lower concentration of oxygen in the blood. And you can see as a consequence, you see there's a red arrow there because those red oxygen molecules there are moving from the alveoli into the blood. The carbon dioxide, on the other hand, which would have been produced by respiration, and there's be a high concentration of carbon dioxide in the blood when that blood is brought back to the lungs. It's at a high concentration in the blood and a low concentration in the alveoli. So you can see that that arrow, that grey arrow, is denoting that the carbon dioxide molecules will diffuse from the blood to the alveoli. Now, it's worth mentioning that it is possible to speed up diffusion to make it happen faster, and there's two ways of doing that. The first way is to increase the temperature, because that literally makes molecules move faster. And because we're talking about the spreading of molecules, just increasing the temperature means they'll move quicker. And the other way is making that concentration gradient higher. Now, in the body, we don't really see a change in temperature that would affect diffusion, but we do see a manipulation of the concentration gradient. We do see situations whereby we try and increase the concentration gradient, either by getting a higher concentration on the side with, in this case, the oxygen, so trying to get more oxygen on this side, or by getting a lower concentration of oxygen on the blood side. And that increases the difference between the two sides. So we say it increases the concentration gradient and it makes diffusion happen quicker. Right, as I've done with you before, I've got here some blanks for you to fill in. So this is a good opportunity to pause the video, see if you can come up with what those words are, and it just gets you involved a little bit more in the process. And then we'll go through them once you've had a go and you can restart the video. Okay, so what should you have on here? Well, diffusion is the spreading of particles from an area of higher concentration, is number one, to an area of lower concentration. We say the particles move down a concentration gradient, is number three. Diffusion is a passive process. I've picked out, if you look at the words I've picked out here, the idea of high to low concentration is key. This idea of a concentration gradient is key. I'm choosing the words to leave out. The fact that it's a passive process, important to pick that out because it doesn't require energy. It's important to pick it out because we're going to see an active process, a non-passive process shortly with active transport. Two of the most important substances that enter and leave cells by diffusion are oxygen, needed for respiration, and carbon dioxide, which is a waste product of the process. We could also have referred to photosynthesis, in which case oxygen is the waste product and carbon dioxide is needed. Speed of diffusion increased by increasing the temperature, because that makes particles move faster, or increasing the concentration gradient. That is the difference between high and low concentrations. Okay, there's our starting point. Okay, diffusion. Here's a little task. I just mentioned key processes, respiration and photosynthesis. Based upon that, you can have another pause of the video and decide whether you should put in or out of the cell for oxygen, the name of process which needs it, and the name of the process which produces it, and do the same for carbon dioxide. Okay, so the focus here, I have to say, the bias here is towards human. So I'm saying it's diffusing in to the cell because respiration is taking place inside cells. And if respiration is taking place inside cells, that's going to cause the concentration of oxygen inside the cell to decrease. And that's going to mean there's going to be a higher concentration outside. So it's going to move in. strictly speaking, if we were talking about photosynthesis, we could say it the other way around. It would be diffusing out of the cell. So this last one, name the process which produces it, is photosynthesis, but it's plants only. So really, respiration is a better focus point because it happens in all organisms. And it's just going to be the opposite now for carbon dioxide. A name of a process which needs carbon dioxide is photosynthesis, and the process that produces it is respiration. And again, I'll say there's a bias here that in and out that I put on there really are relating to respiration. Okay, when respiration is taking place, carbon dioxide is being produced inside the cell. So there'll be a higher concentration inside the cell. So carbon dioxide will diffuse out of the cell. Now, active transport is like diffusion's cousin. There's a few parts of active transport which are distinctly opposite. So here's one, active transport, movement of molecules. Now we're going in the opposite direction here. It's going from a low concentration to a high concentration. So whereas diffusion was high to low, active transport is low to high. And therefore, what we're seeing is, look, you see inside the cell, we've got a low concentration. We've got fewer green molecules here. Outside the cell, we've got more green molecules there. So we've got a low concentration inside the cell, a high concentration outside the cell. And that, whenever we see that, that's active transport. Now, the problem with that is that those molecules don't really want to go that way. Molecules really want to go to the easiest path, which is the diffusion path, because it's passive, it doesn't need energy. So those diffusion molecules, those green molecules would actually rather be moving from out of the cell to inside the cell. But the cell membrane in this case is stopping those molecules moving. This is not oxygen diffusion. Oxygen diffusion cannot move by active transport because they move quite happily, freely through the cell membrane. This sort of layer of red and then a bit of yellow and then another layer of red is meant to denote the cell membrane. And those green molecules are not able to move across the cell membrane. If they did, they would just diffuse across, but they can't. In which case, the only way you're going to be able to get those green molecules out of inside the cell, so out of the cell and into the exterior, is to use energy to pump that substance, and it means going against or up the concentration gradient. Okay, so key features here of active transport: it uses energy. I mean, its name is active transport. Active is the opposite of passive. So by definition, the fact that it's called active transport tells you it's transport needing energy. The term active means it needs energy. So those molecules are pumped against the concentration gradient from low to high. And this brown thing here, this brown, what looks like a brown blob, that's actually a protein molecule, a big protein structure, which is responsible for doing that pumping of those green molecules against the concentration gradient. They need a dedicated transporting protein which does that. Okay, right, let's try this. Little bit of a recap and also a little bit more to do with active transport. Same thing, please pause the video, have a go, and then restart when you're ready. So hopefully you've done that. So it says diffusion is the movement of molecules from an area of higher concentration to lower concentration. This is what we've seen before. They move down a concentration gradient. Yep, great. Diffusion is called a passive process because it does not require an input of energy. Active transport now is the movement of molecules from an area of low concentration to high concentration. It's the opposite. And in order to do this, the cell has to use energy, that is what active means, to pump the substance up or against the concentration gradient. It's great having both diffusion and active transport on the same slide there because you can see how they differ. You can see where they differ. Many parts of those sentences are identical. It's just certain things have flipped around. Right, worth having a look at a couple of questions here. So as always, so you're welcome to pause the video and have a go at these. But for now, we'll so please do so. Now I'm going to go through these questions and we'll see what you've come up with. So the first one actually is a bit of a recap on the structure of the cells. It asks you to identify different parts of the cell. The reason I put this bit on first is, and a lot of people do struggle with knowing what goes with what A, B, C, and D are. And it's, look, remember the key process that's going on inside the cell is respiration. That'll be happening more than anything else. So that tells you that oxygen is being used up and carbon dioxide is being produced inside that cell. Okay, so that's why I've put that on there. You're welcome to pause the video again if you want and have a go at those questions on the bottom, because that might be a useful clue. For now, though, let's go back up to this question 1. So the different parts of the cell, we've got a cell wall, we've got a cell membrane, we've got a cytoplasm and the vacuole. Very basic question there because it gives you the words that you need as well. But this is why I've put this question on there, because it does ask you to identify which is the movement of oxygen, which is the movement of carbon dioxide. So oxygen is A. So how do we know oxygen is A? It's because we've shown a high concentration on the outside and a low concentration on the inside, and we've got oxygen, and we've got this arrow moving in. So the first thing, we know that this is diffusion, and we know that oxygen moves by diffusion. We know it's diffusion because it's going from a high concentration to low concentration. It could also be B, that's going from a high concentration to a low concentration. It couldn't be C because that's going from low to high, and it couldn't be D because it's going from low to high. So it's A or B. But the key thing here is that oxygen is being used up in the cell, and consequently that means that it's going to be a low concentration inside. That makes movement of oxygen to be A. But it makes the movement of carbon dioxide therefore has to be B, because carbon dioxide is being produced as a result of respiration inside the cell. So it's at a high concentration and it'll be relatively low outside, and therefore carbon dioxide moves out. And the process by which these gases move, there's diffusion for you. Okay, C and D would be active transport because it's going from low to high concentration. Right, another question here. It asks you to, again, you can pause and you can have a go. Just give you a moment. There it is. So let's see how you get on with these. So the first thing was, again, is a cell question. I ask you first of all to complete the drawing of the plant cell. Loads of students get this wrong because they are ready to make this the outside of the plant cell, but that they label that the cell membrane. So actually the way that should be drawn is we put the cell wall outside, a line outside that. Forgive my drawing, it's not the best, it's just the nature of trying to draw on a PowerPoint slide. So the line drawn outside the cell membrane for 1 mark. You need to have the nucleus, a vacuole and chloroplast, large vacuole there, chloroplasts, correctly drawn. I must be able to identify which is which, so I've shaded in the nucleus to make it look different. The vacuole is big, and 3 smaller ones, which must be the chloroplasts. Function of the cell membrane controls what comes in and out of the cell. And now we get on to some questions on diffusion. So it shows 2 cells which are carrying out respiration. So we're switched on to, right, oxygen being used, carbon dioxide being produced. It shows oxygen molecules on the outside. And it asks now in the following straightforward question, tick the box, so in cell A, the oxygen molecules move. Do they move into the cell, out of the cell, or there's no net movement? Net movement means overall movement. Okay, so that's going to have to be A because it goes into the cell. I've got a higher concentration outside. Cell B, I've still got the movement into the cell. It's a higher concentration outside. But in which direction would be the greater net movement? Well, there we're looking at the concentration gradient, and you can see that I've got fewer oxygen molecules inside cell B than I have cell A. And it looks like the concentration outside cell A is pretty much the same as the concentration outside cell B. So our outside concentration is the same on cell A and cell B, but our inside concentration is lower on cell B, suggesting that there's a greater concentration gradient in cell B, and therefore we're going to have the greater net movement. And in the process which oxygen molecules are moving, that's diffusion again. Here's one more question. Now, this is focused on active transport. Again, you can pause it. And let's go. So the method used by the root hair cell to take up nitrate from the soil. Right, so the key things to focus on here is look what it tells you. Okay, you've got a vacuole containing a high concentration of nitrate and soil water containing a low concentration of nitrate. So if we're going to take up nitrate from the soil, it's going from low to high, and therefore it has to be active transport, or you can say active uptake. And then it asks you, it says gardeners dig soil to let more in. Explain why this increases the uptake of nitrate by root hair cells. Now, this is the first question that we've had then, which really asks you to do a little bit. 2-mark question, and you've got to write rather than tick a box or just label something. Right, so the key thing here is appreciate that active transport needs energy, and energy comes from, was released as a result of respiration taking place, and respiration, or rather aerobic respiration, needs oxygen. So more air in means more oxygen. So there, oxygen is needed for respiration. What I've done with highlighting is sort of saying the things I'd expect you to say. So you wouldn't say active uptake, you said active transport. And the oxygen is needed for respiration. That's the likely thing for you to say. But you could also say to release energy, you could say to make ATP, yeah, fine. But oxygen needed for respiration is the most likely thing. That's a very brief answer to get 2 marks. But remember, always give plenty of information because you never know what's going to be on that mark scheme. So don't keep your answers brief because you don't know what's going to be there. Right, okay. So let's go back to what I said we were going to do. We were going to explain diffusion, the role of the cell membrane diffusion. Done that, explain the active transport, and now we're on to these last 2 to explain osmosis as the diffusion of water and explain the term selectively permeable membrane. Okay, now we're going to take a step up here because osmosis is the more tricky of the 3 modes of transport. I cannot stress enough the importance of learning the definition for osmosis. Okay, here we go. You hear various different ways of defining osmosis, but I would strongly encourage you to use this version here. So osmosis is the net movement of water. So yeah, so maybe I'll just rephrase that. You can say it's the movement of water from high water concentration to low water concentration. You can say that, okay? I would discourage that. And the reason, and it's often seen on mark schemes, and it's, it's fine. But the reason I would discourage that is because it's very difficult for us to gauge. We don't really have a measure of water concentration. We can measure what's dissolved, which we call a solute. So we can measure the concentration of what's dissolved in water, but we don't really measure what water concentration is. So questions rarely, will never say about water concentration. There's no scale for measuring water concentration that we use at GCSE. But we do have a scale for measuring solute concentration, or the concentration of something which is dissolved. Now we're always going to have to say this third part. I said about learning this definition. So this is fine. This is an acceptable definition. Osmosis, the movement of water from high water concentration to low water concentration across a partially or semi-permeable membrane. I prefer to say that osmosis is the net movement of water from a dilute solution to a concentrated solution, again across a partially impermeable membrane. Now you can learn either. You've got to learn both those things in black. You've got to know it's what comes in between, what's within that sandwich, that you can choose which one you want to learn. I often wonder whether it's worth, I often say to my pupils, you can choose either of these, and then I tell them I think you should be saying dilute to concentrated, but I give them that option. Saying water concentration is not a problem. When it comes to mark schemes, you'll get credited for that. But I think it's more suitable for you to use dilute and concentrated. And the main reason I say that is because most students know what dilute and concentrated means because they are familiar with making a squash at home. So you know what dilute squash is, you know what concentrated squash is. Concentrated, you've got loads of squash molecules compared to water, and dilute, you've got few squash molecules compared to water. You could say dilute, you've got too much water in there, and concentrated, you haven't got enough water in there. All absolutely fine, but you understand that. So I always encourage pupils to use the term dilute and concentrated. So here you have an example really where this actually works okay for water concentration, because we can see this. This is showing the red is meant to denote water molecules here. So I've got side A, side B. So sorry, let me rephrase that. The red is meant to denote dissolved solute molecules, so substances which are dissolved in there. So it doesn't matter what it is, say red is a glucose molecule or a salt molecule, and those glucose molecules, the red, are dissolved in the water on this side, and the those glucose or salt molecules are dissolved in the water on this side, and the dotted line is meant to denote a partially permeable membrane or semi-permeable membrane. You can say either. And semi-permeable or partially permeable means only some things get through, and specifically it's only small molecules that can get through. And the way it's shown there is quite good, actually. It's spot on. See those red molecules? Can you see the gaps in that membrane? Those red molecules can't get through there, so they're going to be stuck on the side that they're on. Water molecules, though, are very small, and water molecules are able to get through the gaps in a semi-permeable membrane. So water is free to move from one side to the other. But the dissolved particles here, the red ones, are not free to move because they're too big. It's a semi-permeable membrane. Now I've labeled this left-hand side as a high water concentration and the right-hand side as a low water concentration, but it didn't really work. It's high water concentration because there are fewer dissolved molecules on there, so it's relatively more water on the left-hand side and relatively less water on the right-hand side, but I don't like it. Better to refer to this left-hand side as a dilute solution and the right-hand side as a concentrated solution. I think you understand that a lot better, so that often resonates far better with students. So what happens then to water? Well, water will move from a dilute to a concentrated solution. That's what I've got there in green at the top. So I know then, once I've got this definition, all I need to do with an osmosis question is figure out where is the dilute solution, where is the concentrated solution. And I often encourage people to draw an arrow to write on each side, because there'll always be two sides of a semi-permeable membrane, a dilute solution, and the other side a concentrated solution. And that's the only bit you've got to figure out. Once you've done that, you can draw the arrow because you know it goes from dilute to concentrated. And the reason you'll always know that is because you've learned the definition. So you've learned osmosis is the net movement of water, and I would suggest from a dilute to a concentrated solution across a partially permeable membrane. There we are. That's what would happen if you gave that a bit of time. The water would move from the left to the right, and therefore you'll actually see an increase in the height of that water on the right-hand side. Okay, and this partially permeable membrane, just to make that clear, that would be like a barrier that would run right the way through. It looks like it's just this line drawn on the edge here, but it would go all the way up from top to bottom and right the way across that what looks like a upturned beaker, doesn't it? Look like there. Right, okay. Well, what effect does that have on cells? So if we take an animal cell and a plant cell and put it in a concentrated solution, that would mean that inside the cell, animal or plant cell, is more dilute, and therefore water will move out of the cell and that cell will shrivel up. So the normal red blood cells, which normally look full, water moves out. When they go in a concentrated solution, they become all shriveled up because inside the cell is more dilute to outside when placed in a concentrated solution. So loads of stuff dissolved in this solution outside. So inside would be relatively dilute. In plant cells, you can often see that the cell membrane pulls away from the cell wall. So it's shriveled up, and that cell membrane, which is usually butted up against the cell wall, is now pulled away from the cell membrane, and we call that condition plasmolysis. If we do the opposite and we take cells and we put them in pure water, then those cells will swell. So placed in a dilute solution like pure water, you cannot get a more dilute solution than pure water. That means inside the cell now is relatively, compared to the outside, is concentrated. So water moves into the cell, and those cells will swell. And with animal cells, they will eventually burst, because that be like just blowing up a balloon or filling a water balloon and filling it and filling it and filling it. And there comes to a point where the wall of that water balloon can't take it anymore and it will burst. With a plant cell, they don't burst because they have a cell wall. So what happens with a plant cell is they start that contents of the cell will swell in the same way as an animal cell, but once it starts to push against the cell wall starts to push back and restricts how much that cell can expand, can swell. And so plant cells don't burst. They get to a stage which we call being turgid. And that's actually a very healthy situation for a cell. Plant cells want to be turgid. They certainly don't want to be plasmolyzed, having undergone plasmolysis. They want to be turgid. So much better. That's actually what you'd expect to see with a plant that's looking healthy, turgid cells, and it would look normal. If you have plasmolyzed cells, I mentioned turgid there, if you have plasmolyzed cells, that plant would look weak and shriveled, and the correct term for that actually is wilted. Okay, give you a couple of moments to have a go at, you can pause and have a go fill in the blanks here. Okay, this is all the key information with osmosis here. The net movement of water from a high, well here I've got water concentration to a low water concentration across a semi-permeable or partially impermeable membrane. I don't like that though. I prefer that it's the movement of water from a dilute to a concentrated solution, and you will see me using that throughout now with any past paper questions. When put in a concentrated solution, the cytoplasm of both animal and plant cells will shrivel due to loss of water and the cell may die. In plant cells specifically, when you do that, cytoplasm may pull away from the cell wall, a condition known as plasmolysis. We say the cell has plasmolyzed. If an animal cell is placed in pure water, the cytoplasm inside the cell will always be more concentrated than the water inside the cell. That makes sense, doesn't it? Inside the cell, you're going to have some stuff dissolved in it. So it will never be as dilute as pure water. So if pure water is on the outside, we know that the outside will be more dilute than the inside because it's pure water. You cannot get more dilute than pure water. So water will continue to move in, and eventually the animal cell will burst. Plant cells don't do that. They don't burst because their cell wall stops that continual swelling. And when it's full of water, we say it's turgid. Right, okay. Quick look now, which direction is water going to move? So you can see that if you got, I'll go back one, if you see this situation here. So with this, you can, well, I don't know, I still wouldn't be referring to water concentration. I say it's more dilute on this side because I've got fewer solute molecules, more concentrated on this side because I've got more solute molecules. Water moves from dilute to concentrated. If you go the other way, can you see, or rather, can you see that I've got 5 molecules on this side, solute molecules, dissolved molecules, red ones, and 5 on this side. So I've got it even on both sides, and what you have is no net movement. You will have water molecules moving in and out of that cell, but there'll be no overall movement. For every one that goes in, there'll be one that comes out. And finally, we've got that opposite situation. I've got a low dilute solute concentration, dilute solution on the right hand side, concentrated on the left, and therefore water molecules move from dilute to concentrated. Right, okay. We're going to look at a number of past paper questions now, which are all pretty similar, actually. I'm going to give you a chance to pause and have a go, and then we'll go through these questions. Okay, so the first one here is quite nice. It's a foundation question. It's just fill in the blanks, and they give you the words. So mark schemes tend to favor this idea of high water concentration to low water concentration. But I still maintain what I say, that it's best to understand it with a dilute to concentrated, because you'll see why shortly. So during osmosis, water moves from a region where it is at a high concentration to a region where it's at a low concentration. So the main thing to avoid here, the thing you really can't do, is ever talk about movement. When you're talking about osmosis, you cannot say water moves from, be careful how I word this, but you cannot say that osmosis is happening and it's moving from high concentration to low concentration, because you need to make it clear that it's water molecules that are moving from high concentration to low concentration. And I would always penalize pupils when I'm marking if they don't say here, so water moves from a region where of high water concentration to low water concentration. If they don't say the water on there and there's any ambiguity, I don't give the mark, because that looks a lot like diffusion, and this is not diffusion. This is a special case of diffusion with water involved. So if you're going to talk about water concentration or high to low water concentration, make sure you include the word water. And then saying it's through a semi-permeable or partially impermeable membrane. Now we've got a situation here where we've got potato chips, lengths of potato, which are put into blackcurrant squash of different concentrations. So, well, 0 concentration of blackcurrant squash in one is pure water, a weak solution of blackcurrant squash and a strong solution of blackcurrant squash. And they try and help you here. It's a foundation question to try and let you focus on, well, where is they want you to use this, or they know you may be using this idea of high water concentration to low water concentration. So identifying which of those tubes has the lowest concentration of water, identifying that as being 3, because you have got the strongest solution of blackcurrant squash, and 1 has the highest concentration of water. Again, I don't like the terms talking about concentration of water. I think it's misleading, but when you're forced to like this, then it's pretty straightforward that pure water must have the highest concentration of water. Right, and then we've got these another bit to do. So again, you can pause the video here and have a go. So they say after 30 minutes, they removed the potato chips and recorded the mass of each, a very, very common experiment, recording the mass of potato before and after subjecting them to different concentrations of solution. If it's not mass, it's length, and we'll see some of those in a moment. So it asks you then, number 1, use a bar chart to complete the results table above. So you can see the black is mass at the start, so you just need to read those off, and they're all the same. Is 20 you read across, and then complete the bar charts for tubes. So 20 for these, and then we can do the mass after 30 minutes here. That's 24. Be careful. People might well say 23 because it's 2 squares down from 25, but each square is worth half. So 2 squares is worth 1 full unit, so it's 24 there. Complete the bar chart for tubes 2 and 3 because it gives you the 20 and it gives you the 15. So I've rather crudely drawn these bars on here. You can see a tube 2, there's my 20. The key thing is at the top where that line is drawn, and tube 3 is 15. You see that line's drawn there. Not very good bars, I appreciate that. And state the number of the tube in which the concentration of water in the chips was the same as that in the solution, and give a reason for your answer. Well, it's tube 2 because in tube 2 there's no change in mass. So we didn't get movement in or out of the potato, and therefore that means that that concentration of squash must be equivalent to the concentration of those things that are dissolved inside the cell. So there's no overall movement. How do we know there's been no change in mass? It was 20 grams after 30 minutes. And why the potato chip in tube 1 gained mass? Look, it gained mass. Well, that potato was put in pure water. And so water concentration was higher outside the chip, or you could say it's more dilute outside the chip, and water passes in by osmosis. The chip gains water then. Okay, right, another question. So I'll give you a little bit of time to familiarize yourself with what's going on here. Pause the video. And now hopefully you've done that. And now we're looking at something very similar. So we've got potato tissue, so potato chips, cylinders that are put into different concentrations of sugar solution. Okay, lower sugar solution 0.1 to higher sugar solution 0.5. This is a very common experiment that you probably did in class. And then those the lengths were measured at the start and after 30 minutes, and the graph shows the changes in length. So we can see if we look at the 0.1 sugar solution, the potato that was in there, it increased in mass. Look, it's the change in length. Change in length there, so it's increased in length by 2 millimeters. The one that was in 0.1. The one that's in 0.2 increased in length by 1 millimeter, and the one that's in 0.3 moles per decimeter cubed concentration, that didn't increase or decrease. And the one that's in 0.4, this one up here, that decreased by 1 millimeter, and the one that is in 0.5 decreased by 2 millimeters. No question on this yet. It's a good idea when you get to a graph to try and figure out what's going on. Okay, to try and figure out what's going on. And then, so let's look at what the questions, you'll come back to that in a moment. So what concentration of sugar solution is the same as the concentration of the cell sap in the potatoes? Well, look, 0.3 cause there to be no change in mass. So the answer there to that first one is 0.3. And it tells you to explain your answer. Well, there's no change in mass. That's all you need to say there. They're giving you 3 lines. There's not much to do. And then it asks you what would be the expected change in length in sugar solution at a concentration of 0.25. So I'm going to go back to this graph here, and that's why I've drawn this line on here. Here's 0.25. I read up. I read across. The expected change in length would be an increase in length of 0.5 millimeters. So I go back to that. There it is, 0.5. Right, now here we go. Here's our stock answer. Explain the change in length of potato cylinders when they were placed in a sugar solution of 0.5 moles per decimeter cube. Just go back down here. So what's happened? Those have decreased in length. Okay, so now I need to start explaining what's going on. And this is a standard stock answer. It's commonly worth 3 or 4 marks. The first thing we have to say the way in which water moves. The next thing we have to give a direction. So I'm going to say it's going from the dilute solution, which was inside the potato cells, to the concentrated solution, which is outside the potato cells in that sugar solution. And then I'm going to say across a semi-permeable membrane. Now, everything that you see in that answer, other than what's inside the brackets, absolutely you need to learn that. And whenever you get a question on osmosis, write it out. And then the only thing you've got to do is figure out what's going on inside those brackets. Okay, now that's not necessarily that easy. But on a 4-mark question, you will get a mark for saying that water moves by osmosis. You will get a mark for saying that it's across a partially permeable or semi-permeable membrane. There's a good chance you'll get a mark for saying dilute to concentrated, okay? But you are going to need to say which direction that water moves, okay? So by saying it's dilute to concentrated, saying it's going from inside the potato to outside. And that's why, if I go back here, that's why the potato lost mass, because water moved out of the potato. And water will always move from a dilute to a concentrated solution. Okay, we're going to see this setup many times. We can also say this, high water concentration to a low water concentration, but see this concentrated sugar solution of 0.5 you've got to think of as being a low water concentration, and I think that's a hurdle that pupils find difficult, so I stick to what's written at the top. Okay, right, another question. Again, you can pause, see what's going on. So in this case, look, it's the same thing. I've got potato placed in water at each of the following temperatures, 7.5, 25, and 35. In this case, what they did was they recorded the length at regular intervals, 30 minutes for 120 minutes. So the one that was put in 7.5 degrees C, at the beginning, the percentage change in length is obviously 0 because it's starting. So it's at its starting length. Then after 30 minutes, it increased in length by, looks like about 0.5%. And then after 60 minutes, increased in length by about just over 1%. And 90 minutes, it increased in length by 2.5%. And 120 minutes, it increased by about 4%. Okay, so that's what's happening. And then we do the same then for going through the 25 degrees C. It's increasing in length. And the same for 35 degrees C, it's increasing in length. But you can see, of course, it's that the higher the temperature, the faster the rate at which it increases in length. Okay, now, question. Still got it on the background here. Explain why the cylinders increased in length and name the process involved. Here we go. Water moves by osmosis. Same thing as before. It moves from a dilute solution, the water that's surrounding those potatoes cells, to the concentrated solution, which is inside the potato cells. So what I know is water's gone into the potato. That's why they've increased in length. So therefore, I just need to make sure I've got these brackets, what's inside the brackets, correct. It's going into the potato cell, so it goes from the dilute solution, which is the water, put in pure water, and into the concentrated solution, which must be the potato cells, across the semi-permeable membrane. It's the same answer. All I've changed is what's in the brackets. Then a couple of tricky questions here. It says, suggest why at 60 minutes the percentage increase in length of the cylinders at 35 is greater than at 25. Suggest. Well, here I've got the high water to low water concentration again here, but okay, back to this one. So why is it more at 35? Well, this is tricky and something you'll probably will have seen elsewhere. So water molecules, or any molecules, move faster at a higher temperature. So that osmosis process, the movement of those water molecules, will happen faster at 35 degrees than it will at 25 degrees because they move faster. That's really what I'm expecting you to say. Water molecules move faster at a higher temperature. But the cylinders at 35 degrees have reached their maximum length by 120 minutes. How is this length maintained? Tricky question. There's an appreciation that there's going to be a no net movement of water now in or out. So there will be movement of water in and there will be movement of water out. But the thing you're likely to say is that rate of water passing in to the potato is equal to the rate of water passing out. What I've got on here is just a screenshot of what's on the mark scheme. I've highlighted the thing I expect you to say. I think you're more likely to say. Last question. Now we're applying this away from potato cells, and they're looking at the so graph shows the average change in mass per hour of samples of two different species of water snails when they were placed in seawater of different concentrations. So with this snail here, the Potamopyrgus antipodarum, I can't say it either, don't worry. You can see that when this was placed in pure water, so 0% seawater, there was no change in mass, and any increase in concentration of seawater, it then decreased in mass. Whereas this snail here, the Ventrosia ventrosa, when it was placed in pure water, it actually increased in mass. And then we had a concentration of about what looks like about 25%, where there was no change in mass. And then further increases in concentration of seawater cause it to decrease in mass. So let's have a look at those questions. Again, you can pause, have a go. So the rate at which water passes into animals that live in water is equal to the rate at which water is removed. This results in no change in the mass of the animals. Yeah, fine. Use the graph to determine the concentration of seawater in which Ventrosia ventrosa normally lives. Well, this is asking you to say at what point is there no change in mass. So this is where the Ventrosia ventrosa lives, in a concentration of about 25%. That will be its natural environment, a seawater concentration of about 25%. So sort of a mildly freshwater area. So the answer on there is 25%. That's the concentration in which it would live. Name the process which causes the other snail to decrease in mass as the concentration of seawater increases. Talking about this other snail here, look, no matter what concentration of seawater, it decreases in mass. Here we go again. Water moves by osmosis from a dilute solution, which is inside the cells, in this case, to a concentrated solution, the seawater. How do I know it's going in that direction? Because they've decreased in mass. So water must have moved out of the cells to the seawater. So I then, because I know that water goes from dilute to concentrated, that's fixed. I can say the dilute solution must be inside the cells and the concentrated solution is the seawater. And finally, don't forget it, across a partially impermeable membrane. If you know your definition, I don't think you will find an easier 4-mark question in your exam. You've got to know that definition and be ready to use it. It says snails that live in seawater can take certain salts into their bodies against a concentration gradient. Name the process. Has to be active transport. And then 2 chemicals needed. Well, really it's asking about what chemicals are needed to make, what chemicals are needed for respiration to occur to release energy. And those 2 chemicals are glucose and oxygen for anaerobic respiration to occur. Okay, that brings us to the end of the cells and cell transport topics that I've covered. So today I've looked at diffusion, role of diffusion, or the role of the cell membrane, looked at active transport, and then we've just spent a whole stint on osmosis, call it the diffusion of water, and what we mean by a selective permeable membrane. And then we've applied that to a load of questions. So hopefully you've got a better understanding. Remember, learn the definitions. It gets you out of so much trouble. Okay. Please recall that we have this live event on 25th of October from 5:30. Please come along, ask any questions, and I hope to see you there. and we can give a little recap of what we've covered, and then it's available for you to check anything out, ask any questions you wish. Thank you for coming today. Thanks for joining. Hope you enjoyed the recording. I hope you got a lot out of it, and good luck in the future.